Inés, ¿nos oyes? Ah, lo puse por WhatsApp. Vale.
Se escucha, se escucha.
Okay, so now then let's start. So it's my pleasure to introduce Professor Albert Antolín from the University of Girona. And he's an expert in machine learning techniques, right, applied to drug discovery. And today I think we have the theory session first, then the coffee break as usual, and then the computer exercise, right, which will be in Teams, but later he will explain it. So, okay, so the first is yours, and thank you for coming, Albert.
Super, thank you so much for the kind invitation. I'm really happy and excited to be here in such a beautiful place surrounded by nature. And thank you all for making it after the late night for some of you. I hope you had a good night sleep. Okay, so today we're going to talk about drug discovery, small molecule drug discovery, and trying a bit to take a step back. I think from what I've heard, a lot of you work in specific techniques or areas of computational chemistry that can be or are applied to drug discovery. But the idea of this session is that you also step back and try to see the whole drug discovery journey as a whole, and also considering the complexity, or this systems perspective. So we will first, I'll be a bit provocative and try to talk through some drug discovery projects. So we learn some of these concepts and try to convince you that drug discovery is not a process. We cannot automate this like a card chain. And at least in oncology, I work in a mainly oncological hospital. I think we need to see this as a cycle, and you'll see a bit why. And I think big data and AI are increasingly important to fuel this cycle and discover drugs more rapidly and efficiently. Particularly if we take this systems approach. Okay, so I'll be a bit philosophical, but I really like this because I think it's really important to understand where we come from and where we are going. I think as scientists, we are very often taught the state of the art, but it's really important to understand how we've come to this state of the art, because the state of the art changes all the time. Particularly if you are already close to AI, you'll see how things are constantly changing. But we need to learn from the past to try to see how we can deal for the future. And we're talking about drug discovery, and I really like to put this in my lessons because I think we need to be humble as well. So we all seem so smart, like using AI and computational methods to discover drugs. But if we go back and see which are the earliest evidence of use of natural products as drugs, we find them in the earliest tombs of humans. And this is not even of Homo sapiens. This is a Neanderthal. And not even just humans or Homo genre, no? Even ants self-medicate. So ants, when they are affected by a specific parasite, they know which natural products, which earth they need to eat in order to cure themselves. So we are not so smart. Even ants take drugs. And this is a recent paper showing how an orangutan finds herbs also to cure the wound that he has made. So, okay, we are not so smart, guys. What's the difference between us and ants? The only difference is to understand how drugs work. Yeah, that's what they don't know. And the question is, do we know how drugs work? So the earliest attempts to try to figure out how drugs work are here in the 1900s. And this is Paul Ehrlich and his earlier visions of how drugs interacted specifically with cell receptors. He had no way of knowing back then if he was right. And in the late 70s, we started to see that drugs interact physically, you know, with proteins, with receptors, and we started to try to design them rationally. Yeah, and this is not a joke. This is not a computational model. This is a physical model. They were building this physically in the lab. And this is the first drug that was designed using earlier versions of talking in the 1990s. Yeah, so it might look to some of you, you're very young, as a long time ago, but if we take this into perspective, it's very recent that we've started to try to understand. And before the late 70s, all the drugs that we were discovering were what we say phenotypic. So we had no idea what the drugs were doing. We just had models, for example, of bacteria, no, that were pathogenic, and we were throwing chemical molecules at them and trying to see if they died, or mouse models, yeah? That's what we call phenotypic drug discovery. Whilst from here, what we are trying to do now is target-based drug discovery. We think we understand how drugs work binding to specific targets, and we try to design them rationally. And again, I really like this historical perspective because it makes us think, or at least understand how we've come to where we are and maybe question some of it, no? So, okay, during the 80s, they discovered that drugs bind to specific proteins, so they started target-based drug discovery. But then in the 80s, some drug, they found that had life-threatening arrhythmias. This is the electrocardiograms. As you can see, this is not good. And after that, between 1919 and 2001, 8 different non-cardiovascular drugs were withdrawn from the market due to this severe side effect. So of course, it was a big concern for pharma industry and you know, healthcare in general. So they tried to figure out what was happening, and what they found is that all these drugs bind to an ion channel called hERG. That is the one causing this severe toxicity. So from this, they concluded that all off-target binding is bad, and that's what you still find largely in research today. And it's very interesting because hERG binding is the only off-target that you are required to provide data for when you're trying to approve a new drug. That's the only thing that you're asked from the regulators, that you demonstrate that your drug is not binding hERG. Again, learning from mistakes. Okay, so how is the small molecule target-based drug discovery today? You've probably seen this in many lessons, this type of linear process where you start identifying a target, then validating it, identifying hits, then leads, optimizing them, having your preclinical candidate, and going to clinical trials. In this kind of perfect, simplified view of how we discover drugs, I want to draw your attention to the off-target profile, because the selectivity of your small molecule that you're developing, it's only evaluated at large panels when you have a lead already you've chosen a clinical candidate. So when you are there and you are in the industry, and I worked in the industry at the beginning of my career, you have a lot of pressure to push the drugs to the finish line. I assure you, unless you hit hERG, or a few other targets that we know that they cause severe side effects, you don't care. So you find a lot of papers in medicinal chemistry that say, we've developed a selective inhibitor of something. If you go back to see the data, very often the drug binds to other targets with different potency that was never removed. Yeah, they just checked and they went to clinical trials or preclinical toxicology. And again, to highlight this is a systems talk. So highlight the simplicity, no? So we try to design specific drugs for only one target, and this is going to solve our disease of interest, no matter how complex our disease is. Okay, so let's go back to the first target-based drug that was a small molecule that was developed in oncology. I don't know if you know about this, but imatinib, it's a very famous drug. Again, for those that know a bit more of oncology, the chronic myeloid leukemia, which is the disease that this was originally developed for, has very specific architecture or genetic architecture in the sense that it's driven by a translocation. So two parts of a chromosome come together and they create this fusion protein that is overactivated. Yeah, and that's what drives the disease. This, I can tell you, it's not a general mechanism in most cancers that we have today, and they are concerned that are much more complicated. But here with this translocation, you have overactivation of a single kinase. So what scientists at Novartis thought, could we block this kinase activity to stop CML? Okay, they did an in-house screening. Actually, the hit came from another program of company. And as you probably all know, they started to optimize this hit, as you can see, growing the molecule through here and here. Then they started to hit some, in the hit to lead some metabolism problems. So they methylated this position. This is likely because it's a site of metabolism. And then they grow the molecule. Normally, the introduction of these groups is to improve pharmacokinetic properties, etc. And they discovered imatinib. Yeah. So from discovery, sorry, I can't see very well. So from discovery of the lead to the first approval in CML, 9 years. Okay, so again, pretty standard, really amazing. And the impact of this drug in the treatment of CML is amazing. Yeah, so it went from, I don't know, very high levels of death to very high levels of control. Yeah, so this was an incredible success. Then, I didn't say, but so my idea is that in 2 or 3 points in this presentation that you can play a bit as well with the data and don't believe all I'm saying. And this is one of them. This way is not so boring, hopefully. So the question is, okay, so Novartis scientists try to develop the selective inhibitor of ABL1 kinase because you saw the translocation and it's driving in CML. The question is imatinib so selective? You probably all know that, but you can check selectivity data in Campbell, or for example, in cancer, that it's the database that I contributed to build during my postdoc in the UK. So I give you 10 minutes or 5 minutes maybe, if you can check and tell me if imatinib is really selective for ABL1 or not. Sounds good? If you have any questions, you can raise your hand or ask. And of course, there are plenty more databases that you could check. It's quite mobile friendly.
Potevi aver visto battere per no, perché ci metti al momento, quando sei deciso, però la igual persona. Pero hay más de una vez Antolín, así que igual te. Vale. Y me gusta esa Barcelona. Sí.
Okay, so can we do a small poll? Who thinks imatinib is selective? Could you raise your hands? Don't be shy. Who thinks it's not selective? And the rest are sleeping. Okay, let's demo this for you. You're right, obviously. This is cancer. I'll promote this a little bit because it's the one I was involved in. Actually, cancer, as you'll see in a few slides, incorporates Campbell. So you're basically searching the same data. Cancer has a few more, but. So Campbell has mostly, as you see, when you search, then you have to click the thing that you're really meaning to search. So Campbell has more bioactivity, pharmacological, and chemical data. Cancer is a bit more broad, as you'll see, and it has a lot of more cancer and biology data. But in terms of the pharmacological and chemical data, as I said, is quite similar. So what we try to do in cancer is that when you search for something, we give you kind of summaries of all the data available, and then you can dig deeper into the data, no? So here, for example, you have different types of data. And if you go to bioactivities, you have summaries of all the data that you have. Of course, you have a lot of data points. So maybe if you looked at Campbell directly, you had all the different, I think I saw some of you in your screens, you had all the different bioactivity data points, yeah? Here, if you scroll to the bottom, we have this, what we call target activity profile, which is trying to give you, it's now not sorted anymore. No. It tries to give you a summary, yeah? In any case, if you sort this by potency, for example, yeah, you'll see. Well, the problems of doing live demos, they've changed this a little. But you'll see that it binds a lot of proteins. Not all of them are kinases. You see carbonic anidrases, etc. So yeah, imatinib is not completely selective. And you'll see, oops, how can I go back? Yeah, so here you are. This is the target profile that we had a while ago. It binds ABL1 with high potency, for sure. It was designed to do that. But it binds many other kinases, and as you saw, other proteins that are not kinases even, yeah? So is this good or is this bad? And do you think we still completely know how drugs work? What's these other targets contributing to the mechanism of action, to the safety and toxicity of imatinib? The reality is that we don't completely know, and that's another reality of science, that we don't know lots of things. But imatinib is a very interesting case because imatinib also inhibits quite potently another kinase called KIT. Now, after imatinib was approved, there was a lot of rush to identify new cancer oncogenes and develop new therapies for them, right? And some scientists that were studying a relatively rare gastrointestinal cancer called GIST, they found that KIT was a driver of this cancer. So scientists at Novartis thought, oh, we have a drug that, yes, binds ABL1, but also binds KIT with quite high potency. Could we develop it to treat this other cancer? So they did it, and they managed to approve it, to show benefit for this type of cancer as well. And then, well, I don't have it here, but it even was approved for another type of cancer driven by PDGFR translocations. So one drug, 3 different indications due to binding to 3 different proteins, yeah? And this is where, well, here you have the selectivity profile. And then here, what it represents is this idea that maybe drugs don't need to be magic bullets, no? This kind of super specific drug, but they can be magic shotguns or a shot that you can hit several targets and, you know, be more effective or show efficacy in different types of cancer or disease. This is what we call polypharmacology, the binding of a drug or a small molecule to several targets. And it's good or it's bad. So here you have another example from antipsychotics. So this is a very influential review. And actually what they showed is that so antipsychotics had very severe side effects. So all the antipsychotics that had been developed before target-based drug discovery, they were effective, clozapine, etc., but they had some severe toxicities. So some companies tried to develop very selective antidepressants for specific GPCRs. What happened is that they were not effective. Yeah. So polypharmacology can contribute to efficacy, but of course, see how promiscuous these antidepressants are. This is obviously not good either. They produce severe side effects on these patients. No? So what I think is that we need to find the right balance. And actually what happened is that after imatinib, there was a bit excitement in the oncology field to try to develop non-selective kinase inhibitors to try to approve them for lots of indications. And you had a few of these multi-kinase inhibitors. This is the kinase 3. Each dot is a kinase that is inhibited. Look at sunitinib, for example, or sorafenib, how many kinases they inhibit. The problem is that these drugs are relatively toxic, even for oncology. So they managed to approve them for some indications, but for others, it's too toxic, it's not sufficiently beneficial, yeah? So this idea of just hitting everything doesn't work anymore, yeah? So what I think is that we need to find the right balance. That might not be complete selectivity, but also it's not broad multi-target activity. Okay, another example. So here you have vemurafenib. This was discovered from a fragment, as you can see, fragment growing. It's another technique widely used today. And again, the idea was to find inhibitors of a specific mutation of a kinase. It's BRAF V600E. And as you can see, they managed to do that by growing the molecule, etc. But if you look at this, again, despite having been designed from a fragment, growing it to make it perfectly selective for a specific mutation of a protein, it does bind to many other kinases as well. So it's not completely selective either. So one big problem we have in oncology, and here is where we'll come to this idea of the cycle of drug discovery, is resistance. And I'm sure you are all aware of this, because sadly, we've all seen someone close to us that has cancer, starts to respond to treatment, things go well, and then after a few months or years, the cancer comes back more aggressive. That's what we call resistance. So here you see, and this is a bit to see the potency of these targeted therapies in oncology, but also the limitations that we face. Now, this is a patient that presented in the clinic with metastatic melanoma. All the black masses that you see are metastases. After 2 weeks of treatment with vemurafenib, most of the masses are gone. The recovery of those patients, it's amazing. Like oncologists were super surprised, yeah? I remember a picture where you could see physically also the tumor masses gone, the color of the screen changed, amazing. But sadly, 6 months later, the cancer is back, more aggressive, more difficult to treat. So one of the strategies we are studying more comprehensively now is trying to combine targeted therapies. And there are different theories, like one theory is that when you block a specific pathway, there's normally crosstalk to other pathways. So if you block another pathway, you see improved response. This is the situation now with melanoma. For example, a combination between BRAF inhibitors and MEK inhibitors is approved. So you can block these two parallel pathways and you see improved responses. But there are other theories, and again, we don't completely understand how this works in patients. Tumors are not homogeneous, they are generally heterogeneous. So some argue that what drug combinations are doing is really targeting different subpopulations within tumors. There's even more provocative views that I quite like, saying that drug combinations simply work because some drugs work for some patients and other drugs work for other patients, and they are not really working together in the same patient. Again, we don't completely know. A lot of research going on here, but it's difficult. I don't know if I have the slide here. Yeah, but it's difficult. Context is really important. So you saw the results in melanoma. This is colorectal cancer, the same mutation, but BRAF inhibitors don't work because just this cancer is much more complex and the cells are engineered in a different way. So we cannot always kind of take the learnings from one context and apply it to other contexts. And here, just to give you an idea of how we are pushing this, this is a combination between three drugs, an EGFR antibody, a MEK inhibitor, and a BRAF inhibitor. And it works, it's approved, but the increase in survival, it's 9 months. And this is the sad reality in oncology, that some of these combinations or some of these therapies that we are designing just push a few months the survival. It's a bit tricky. I'm not completely against this, because you also need to understand that some of these therapies are tested with the patients that don't have any other treatment available. These are very complex metastatic disease. Very often, when a combination, for example, or a new drug is approved in this setting, then it's studied, give it earlier, or give it earlier in combination with the standard of care. And these numbers improve a little. But the reality is that due to resistance, we are not improving as much as we want. So this is where we and my mentors in the UK thought of this idea of the cycle of drug discovery. Because what I've showed you 2 drug discovery programs, and imatinib also shows resistance, but it's much more minor. So in this sense, imatinib was a strange example because the molecular architecture of the tumor is very driven by ABL1, yeah? But with BRAF mutations, it's very clear. So we identify, it's very clear that BRAF is an oncogene and that these cancers are driven by BRAF, yeah? So we identify a target, we identify hits, leads, we optimize the leads into clinical candidates, we demonstrate that they are safe. We go to clinical trials, we saw a response, and even in those clinical trials, we already saw resistance. So we have to start again and identify MEK as another target and develop the selective inhibitor, you see? And the combination also shows resistance, so we start again. So it's like a circle. It doesn't finish when we go to clinical trials or a drug is approved, because resistance develops quite immediately, yeah? So this is where we see that we need more data, particularly data from the clinic and these mechanisms of resistance, and more AI methods to try to accelerate this and develop drugs more rapidly for patient benefit. So what I'm gonna do today is going through these steps of drug discovery and trying to summarize which computational methods are available, the limitations, no? Often we have the limitations in the data available to build these models, so we need to be aware of that when we use them, and try to give you this kind of broad perspective of where we are in the whole drug discovery and development cycle. So, target identification. I think most of you are chemists like me, and until I went to London, I, like probably many of you, simplified a lot target selection. It's like, yeah, we've identified a protein that's important for my cancer of interest, and we're going to develop the drug for it. And that's the important thing, is to develop the small molecule drug. But actually, from what I've told you and the complexity of it, think about it, like identifying the protein or proteins that are key for your target of interest is probably the most important part, no? Because if you select the wrong protein, your drug's not going to work. It doesn't matter how selective, how, no, the ideal properties it has. So how are we choosing these targets for drug discovery? And again, very often I think things are changing for good, and we are now using more multidisciplinary data and multiomics to select these in more rational way. But target selection, when I was in industry a long time ago, used to be very simplistic. Yeah, this target has been shown to be important for this disease. Let's develop a drug for this target. And you know, we need to do better. We are in a data-driven kind of world now. And it's very important that you remember why drugs fail. So again, today, 90% of drugs that reach clinical trials in oncology fail, and it's quite similar for different indications. If you look at the reasons for failure, these are the reasons for failure. Again, before, like 20 years ago, lack of good pharmacokinetic properties was a big reason of failure. This is going lower and lower. Yeah. So safety, about a fourth of drugs fail because they are toxic, and we'll talk about the data we have and the limitations we have to predict human safety. But look at these guys. Half of the drugs fail because they are not efficacious. Because we select a target or targets, we develop the drug for this target, we go to the clinic, and it just doesn't work. It doesn't kill, or it doesn't show survival, for example, in colorectal cancer, sufficient to get approval. So how are we selecting those targets that when we go to the clinic, they don't work? And it's quite interesting, for example, to know at the cancer genes that we've validated they are important in cancer, and then you check how many we've drugged, and it's a very low proportion. Yeah. Because druggability is difficult, because there are many reasons, yeah, but that's the reality. So when I was in the UK, again, a lot of rain, but we started the Department of Data Science there, and I had the privilege of being there from the start. And some of the idea was, okay, we need to develop drugs. Drugs are failing because of lack of efficacy. So what can we do to improve how we are choosing targets? And one vision was to kind of get all the data and try to distill it. And that's cancer, as you probably see. So one problem we have in drug discovery is that we need a lot of data from different types. It's not just chemistry, it's not just pharmacology. We need to understand the genomics, no? If this cancer is mutated, statistically significant in this cancer, or maybe it's overexpressed. Of course, pharmacology, of course, 3D structures of proteins, but protein-protein interactions, networks, target validation, etc., etc. So the idea was to bring all of these together into one platform. Again, just not to promote only cancer, an alternative that I think is really good is open targets as well. This is a public-private partnership in the UK between a lot of pharma companies and the EBI, and they try to kind of distill as well publications, but also generate their own data to try to assign targets to diseases. If you don't know this resource, I do recommend that you check it. For example, well, of course, if you check for BRAF, you find melanoma is highly enriched. And then our solution, which was cancer. Again, I don't recommend building a database as huge and as multidisciplinary with such different types of data as cancer. It was very difficult to do. It's very large. This is a bit outdated, but it's a relational database, which gives us the possibility of making connections between the different entities in the database. And again, it allows you to do lots of things. I could spend the whole 2 hours talking about cancer, but I won't. Something that I think needs more development, but it's very interesting. It's precisely what we were saying, associating specific targets with diseases, or cancer is very focused on cancer in the biology part or the disease part. So which type of cancer? Again, I think this could be much more sophisticated, yes? But there are some scores trying to help you. If you have a target of interest in seeing which types of cancer would be more affected by this cancer. And the other thing that I think is really cool, and this brings us closer to our topic and chemistry, etc., is druggability. Or how we prefer to call it, ligandability. Because what we are really predicting is not whether a protein, we can discover a drug for a given protein, because to discover a drug, we need to show efficacy. We need to show that this drug works, and we don't know that. What we are predicting is whether a small molecule would be able to bind to a pocket in this protein. So whether it would be ligandable, yeah? So we have different flavors of ligandability, but probably the most interesting is the structural one. Again, I don't have the details, but the cool thing is that, so we developed a ligandability machine learning prediction. It was quite simplistic, to be honest with you. We just fed the algorithm, I think it was a random forest, with descriptors of binding sites of high affinity ligands. Some of them were drugs, some of them weren't. That's why we are predicting ligandability. And basically the algorithm learned what these descriptors were important. Also, for example, pockets need to be of a certain shape. If they are too big, doesn't work that well, for example, need to be quite enclosed, etc. Anyways, I think there are now better methods to do this, but the algorithm worked. And of course, we have a very nice opportunity, or we had a couple of years ago, when AlphaFold was released. Because now we have protein structures of all the human proteome. Yeah? So we can predict druggability across the whole human proteome. And this is just one example. So thyroid peroxidase is the target of an old drug. It had never been crystallized, but we know that the drug inhibited that target, and we do predict a ligandable pocket in thyroid peroxidase. Okay, so now a bit of time for you to raise hands. So how much of the human proteome do you think it's ligandable by a small molecule? So who thinks, I don't know, 90% of the human proteome, we could design a small molecule, has pockets that would be a minimum for small molecule modulation? Who thinks 50?
Less.
Sorry?
Less.
Less. 50. 50%? Nobody buys 50? 25%? Come on, I'm in a room full of pessimists. Okay. 10. 10%? Come on, 5. Okay. Well, you are very pessimistic, guys. But I have good news for you. So our prediction suggests that 42% of the human proteome is amenable for a small molecule. That's a lot, yeah? So if we assume around 20,000 human proteins, it's more than 8,000 proteins. Okay, let's do more of this so you are awake. 42% of human proteins have a pocket that we predict could bind a ligand. How many, so of the 100, you think that we have already discovered a ligand for it? Not a drug, a ligand, yeah? So who thinks, I don't know, that we have discovered a ligand for 40% of the human protein? Yeah, no, that's too much, I agree. 20%. I tell you, you are very pessimistic in this course. 10. 5. 16%. Guys, I don't know, but I'm really excited about this because all of this is an explore. All of this is for you and for me to discover new small molecules for them. Yeah, we have a lot of work to do. Yeah, it seems that AI has solved everything. You'll see in a few minutes my criticism of that, but there's a lot of work to do. Yeah. Now, the last time I asked you to raise your hands, how many of the 100, yeah, do you think we have drugs for? Who thinks 10%? 5%? Now you are too optimistic. 2%. It's 3. Again, all of these, or a big part of this, I think it's opportunity as well. Why? Because a lot of drugs here are, so a lot of targets here are, for example, so I studied a lot on PARP1, which is a protein, yeah? But PARP1 is the most studied member of a long family of 14 members, yeah? Nobody has studied PARP14, PARP16, very few, yeah? But we have ligands for it, so it's here, yeah? But nobody has really tried to develop a drug for PARP14, yeah?
I'm just a bit lost about the difference between ligandability and druggability. So you just explain again what's the difference?
Yeah, it's basically, sorry, go ahead.
Yeah, and why from the 16% that are already ligated, only 3% are considered drugs?
Yeah, yeah. Sorry, I'm very well of raising your hand. Please do this if I'm going too fast because we all have different backgrounds. Druggability and ligandability, basically people use it as synonyms. So it's the same. What I'm saying is that people shouldn't say druggability because practically nobody can predict druggability. Because what is a drug? A drug is a compound that has been approved by a regulatory agency to treat a disease because it has shown in clinical trials that it works. Nobody can predict that. You'll see in a minute, yeah? So everybody that tells you that they have a druggability algorithm, what they really have is a ligandability algorithm. Because what they predict is whether this protein could be modulated by small molecules. In our case, we predict whether the pocket has properties similar to pockets that bind high affinity ligands. Yeah? This is important. What's a ligand? And I probably didn't explain this enough. A ligand is a small molecule that binds to a protein, just that. Here, I use quite relaxed methods. So basically, it means that in Campbell or in cancer, there's a ligand that binds to this protein with an affinity below 10,000 nanomolar. Quite easy because it could, 10,000 is quite weak, yeah? So basically for, I don't know, for part 13, for example, imagine you have a ligand that was published in a paper, no? That had an affinity of 2 micromolar. This is very weak. I don't think it's a good ligand, but it would put part 13 here. Yeah? Drugs are compounds that have been approved by a regulatory agency. So part 1 has drugs approved by the FDA to treat ovarian cancer, for example. So part 1 is here. Yes? Super. So yeah, feel free to raise your hand and ask. It's much more fun and I don't lose you. Okay, so another very interesting thing. Until a year ago, more or less, all my research was on those proteins. Because in my research, I mostly use chemical data, so I'm more cheminformatician, yeah? So we only have chemical data for 16% of human proteins. For the vast majority of proteins, there is no clinical data. Those of you who work more with the structures are luckier because we have much more structures, but chemical data, we don't. You'll see in a moment why I think this is very exciting as well, because I'll talk about some initiatives, and you probably heard others in the news, not like DeepMind, etc., that they are trying to generate data in this space for machine learning models. So I think it's a very exciting time. And this is just to say that, so during my postdoc in the UK, I was in an institute that's a very special place because they also have like, it's like putting a small biotech company inside a public research center. So they also discovered drugs. They discovered 2 approved drugs, which is pretty amazing. But they wanted a system to better select their targets. Yeah. So that's what it was built in the department. I didn't participate in that part, but basically was putting all the data that I showed you that cancer has together to try to help them prioritize the targets that they chose for drug discovery. Okay, and now a bit of time for you. I think I'm gonna do one of these for you, so you see what I would like you to do. But basically, and this kind of ties with the next exercise we'll do after the coffee break. The idea is that you put your hats on as drug discoverers. Yeah, imagine I give you, I don't know, well, it takes a lot of money, but 2 million euros to start a drug discovery project and get to a clinical candidate. Which targets would you choose? Yeah? So I want you to use cancer to try to select targets, yeah? What things could be important? And we'll do it theoretically now in a second, and then I'll show you in a moment, yeah? For example, you need to check ligandability, yeah? Because if a protein is not ligandable, it's gonna be very difficult to discover a small molecule drug for it, yeah? But not just ligandability. You have to check if this drug has already approved drugs. Because if this protein has already approved drugs, you have very strong competition. It doesn't make a lot of sense to discover a new drug unless you have some insider information, yeah? And maybe you want to think as well of failed drugs, yeah? does this target have been pursued in drug discovery? No, so companies developed inhibitors for this target. They took them to the clinic and they failed. Maybe if they failed, you don't want to risk it. Maybe yes. Yeah. I hope you start to see as well that this is not mathematics. Yeah. You need to take decisions with all the information available, but at the end, it has some degree of subjectivity. So let's try. I'll go back because I think I have the solutions. Yeah. So let's use cancer because I think it's the simplest. And I think the first target you had is HSP90. So what would I do? I would go to HSP90. Yeah, it's here. You have to click the right target. And then here at the landing page, you already have quite a bit of information. Yeah. Let me see. So investigational drugs, it means that this protein has clinical candidates. So it has compounds in clinical studies. Not so great if we are starting from scratch. Ignore the chemical probes and ligandability. So ligandability is very high, yeah? So here the chemistry base is very high and I don't know what the structure, I don't know what happened with cancer. No, the structural base we need to enter. And here a structural ligandability, we need to enter to see, yeah? So if we go to explore more, you have for every, they've messed something up. No, it's here. It's there because the Wi-Fi takes a while. Okay. The first chain, the pink one, is AlphaFold. And the others, the red and green, are PDB. Yeah. So all the green chains represent a chain in the PDB for which we detect a ligandable cavity. You don't need to do what I'll do now, but I want to just to show you. Yeah. So if you click one of these, it goes very slowly. Anyways, so you have a way to explore this in 3D. Yeah. So if you select one chain and then you go to chain details. Again, here, for example, you could see it has a co-crystallized ligand. So again, you can figure out that this binds ligands. Yeah. And then it might take a while to load, but you have a way to explore this. You don't need to do this for this exercise or after the break, but just so you know that this is there. Because I think cancer has a lot of data and a lot of people don't dig deeper enough. No, they know these things are there, but they are. Well, let's give it 20 seconds, and if not, I'll go back. But basically, yeah, you can explore the 3D shape of the protein and the different cavities to see which cavity we put in. Yeah, let's forget about this. And the other thing I wanted to show you, if we go back to the landing page. Yeah. Yeah, the clinical candidates. So you can also explore, search by small molecule and explore specific small molecules, luminescence. So I'll talk about this in a second. But something you can check, or I would check as well. So cancer has also the clinicaltrials.gov database here. So if you go to trials, you can check when did the clinical trials started and ended. And you will see that for this drug, there is nothing, no clinical trial ongoing now. In 2020, they stopped. And this compound has been in the clinic from 2007. So what is this? This is a dead clinical candidate. So again, you have to think if you want to risk to develop a new HSP90 inhibitors when at least 2 have failed in clinical trials. Good luck. That's the reality. Yeah. Okay, so I'll put you back this. I'll give you 10 minutes to play a bit with it, and you can feel free to raise your hands, ask questions, whatever you need. So these are the 4 targets that you may want to choose for a new drug discovery project. Yeah, sorry guys. Maybe you could partner with I mean, I now write with an iPad, but I also agree with writing, but it's true, I think that it is now in our field, because everybody knows more.
And an AI tool.
Because we say that okay. Yeah, I mean, I don't know you can calculate I will break over this one the same. Yours up to the walls. So that is because we don't have a review, but they are which I think was on the Texas questo sintomo non c'è un po'. Other design more to push very often we look at like in the next comparisons.
Something that since it advanced and in progress, they run those it's
yeah. The problem is that I don't really know anything about I mean, it's slow. There's a lot of take a lot of risk that you have it. Ah, no, because you're but you have the right one. And the euro last you were looking up so and they don't propose as they are able to get even more for now they are doing they want to do that one. So it's one more. How is it going? You've chosen, you need a bit more time. You've chosen, chosen. Yeah. Shall we do this? Okay, let's do this. So who chose HSP90? Like your perseverance. Who chose big one? Okay. Who chose NTRK1? Who chose then D6A? Okay, not bad. Any other selections beyond the obvious? No? This would be the law. Again, there's no, as you'll see in the next exercise, there is nothing cast in stone. If you want to pursue HSP90, you can. You'll probably have problems finding funding because it's considered a dead target. I don't think it's dead, but so probably the obvious choice would be PIK1, which is what the majority of people selected. And TRK1 already has approved drugs. It's a very interesting target, but it already has, I think, 2 approved drugs. So you'll face a lot of competition. You can do it. And the one that didn't make much sense was the last one, which you all got. That's my choice. I never said you had to choose only one target. Actually, this presentation is about maybe choosing more than one. You'll see why I choose this later. But I think HSP90 is a very interesting target. The clinical target, well, I'll talk about this later because it comes up. But maybe we can design a dual inhibitor that rescues HSP90. That would be my choice. Yeah, and this is a bit where I get a bit more critical about AI and target selection. Okay, so what do I say? So I think we're living very exciting times, but there's always, I'm sure you've seen this many times. So this curve of hype, and then the downside of delusionment, and then the back of the real application. Yeah, so we are probably in the big slope of hype around AI, and people overclaim, and overclaiming is risky. Yeah. And in target selection, you saw what we tried to do with cancer, like put all the data together, but we didn't capture half of the data that's out there. We had lots of problems to put it together. And again, the data doesn't reflect probably well enough advanced disease. Lots of this data is not clinical. Anyway, so benevolent AI, for those who have not heard of it, it's one, or was, but I'm not sure. It's a big company still in the UK. Was one of the first AI in drug discovery companies. It raised a lot of money. It's not here, but huge. They developed a knowledge graph AI to try to identify targets. They ended up identifying TRK as a target for eczema. They developed a pan-TRK inhibitors, went to clinical trials, and it failed. Again, this is N of 1, so we have to be careful. But if you claim that your AI is going to select targets fantastically and all the investors go crazy, and then you fail in clinical trials, well, you are in trouble, right? So I had a lot of friends that went to work for Benevolent and they were fired because of this, because of this failed clinical trial. And again, well, this lesson builds in a course that I teach. So I did talk since 2023. So that's what happened in 2023. In 2024, it laid off more people. And in 2024, it fired more people and went back to just doing AI, because before it also opened drug discovery offices in Cambridge, and then it closed now. So it's very risky to make such claims when we know very little still of how drugs work, of how cancer works, of how patients might respond in terms of safety and toxicity, etc. So I think AI is a very useful tool, and I think we'll come to this, but overclaiming is risky. And I do recommend this review. I think you'll all have these slides, but basically it's very interesting, and I don't want to enter into a whole lot of details, but basically the review argues that AI has shined, for example, in image recognition, but it's a very different problem. One, the matrices of images are complete matrices of pixels. The algorithms have been specifically designed to deal with this data, and you have tons of labeled data to train your deep learning algorithms. This is not what happens in chemistry. We don't know still very well how to represent molecules, which are the best descriptors or graphs or, for different problems. Algorithms are not yet specifically designed for this, and we don't have tons of labeled data. I think there are 3 million chemical compounds known in ChemBot. So it's not much if you compare to all the data that's in the internet, right? And this data is very biased. We don't have very good negative controls, lots of issues, yeah? But when you go to biology, it's even worse. The data we have doesn't represent when the endpoints we try to model, and we'll talk about toxicity a bit later, but we don't have enough good toxicity data, human toxicity, that's what we want to predict. We might have rats or we might have cells, but it's not really what we're trying to predict. The algorithms are not right, so it's very, very difficult, yeah? And here I mean, Andreas is great. If you have the opportunity of hearing him, I do recommend. Again, linking the data we have with the questions we are trying to answer, the representations and the methods, no? And thinking carefully about this. And this is why this is new. I saw this recently. This is a meme I saw in the internet somewhere. But I don't know if you heard, but Demis Hassabis, Demis is amazing. I'm not, he led AlphaFold and it's now a DeepMind Nobel Prize, revolutionary. But he made this claim that AI will cure all diseases in a decade. And this is, not realistic, right? This doesn't help us. And these people were comparing, I don't know, someone at Microsoft then said this, quite a few years ago. It's not true. And this is not going to be true in a decade, yeah? It's not true. The problem is very complex, much more complex than solving the 3D structure of proteins, yeah? We are dealing with human data that we don't understand well enough, and we don't have enough data to model that. So don't worry about these claims. You'll have a lot of work to do in all your careers, I promise. We won't cure disease in 10 years. Okay, target ID. This is the first step. But then we need to identify early chemical matter for the target or targets that we've chosen, yeah? The classical way of doing this is brute force, and still this is the method of choice in a lot of industry, but it's expensive. I have to be honest, more expensive to set up. Once you have set it up, it's not as expensive to run. That's why still pharma industry runs this. Yeah. But there's a lot of issues with the quality of data. They need to validate a lot of the hits, etc., etc. So virtual screening, I'm sure some of you work on this. It makes a lot of sense as as an alternative. And there are very nice approaches coming up or that have been developed. This is one key thing, I think. So we are now able to screen increasingly large libraries. Some of them are known. Some of them are make on demand, what we call. So compounds that we think we could synthesize. And these are billion-sized libraries. So they are developing very smart artificial intelligence method to reduce the computational cost of the screening such large libraries. And now you can do docking or docking guided by machine learning with these methods. An alternative, in case you haven't heard of this, but I think it's also interesting, although I'm very biased, it's something that we call DNA-encoded chemical libraries. These are molecules that are synthesized with the DNA tag, and you add different substructures and different DNA tags. So basically, by sequencing, you can know which compound is binding your target. And again, those libraries are huge, but they also have some problems, like a lot of false positives. So there are successful methods or increasing interest, I would say, in these methods. So training machine learning methods with DNA encoded chemical library data, and then predicting external libraries that are easier to buy to find hits. Yeah. So this is something I'm involved with, and I think this is really going to make a difference to find hits for lots of proteins. The other thing I want to talk about in this kind of hit finding is, again, going a bit against the hype with AI. No? So you've probably seen, Bol3, this kind of AlphaFold-like algorithm now can predict ligand affinity, and it's solved. Basically, you don't need to do docking anymore, and it's all fantastic. And it will find hits for any protein that you like. Well, the solution against this is challenges, computational challenges, and you'll see a bit more about this. No? So if you know the story of AlphaFold, it's very interesting because protein structure prediction has been a big problem, or had been a big problem of molecular biology or biology for a long time. And since, I don't want to get this wrong, but since a long time ago, so for 30 years, there was a competition called CASP for structure prediction, which was run every year, and a lot of scientists participated and tried to make predictions, et cetera, and the field was slowly growing for 30 years. Also, there was a lot of data being put out for free in the protein data bank that people could use to train those models. And that's the ecosystem for 30 years that enabled suddenly AlphaFold to make a jump with the Nobel Prize, etc. But there was a lot of data and a competition where scientists competed in the open, shared their learnings, yeah, and we could assess the methods and compare the methods. Because you've probably seen this, but a lot of papers don't exactly do the same metrics to compare how their algorithm works. All the algorithms look perfect, fantastic. They're all retrospective, no? Which is easier to predict. And then when you use them prospectively, they work much less well, etc. So these competitions, everybody competes, everybody faces the same problem. So you have a realistic view of how all the algorithms work. So these are some sort of challenge competitions called CASH. No. Well, the pointer doesn't work. CASH, yeah. They are run by the Structural Genomics Consortium. They are experimentalists with a lot of experience making 3D structures and making chemical probes, and I collaborate with them. So this was the first challenge that they did. Difficult problems, right? Not a lot of those papers, big papers of AI in hit finding, they find hits for another kinase. That's an easy problem, right? We already know how to find hits for a kinase. They put forward the WDR domain of LARC2. LARC2 is a kinase, but the WDR domain had no known ligands whatsoever. It's a weird domain of this kinase. And they asked people around the world or groups around the world to predict hits. They bought those hits. This was not retrospective validation. This was prospective validation. They bought the hits. They tested them in physical. They crystallized the ones that worked. This is a huge effort. And that's what they found. So 4% of the predictions were right. No? All the algorithms that claim that now hit rates are 30% when you use AI, 4%. And these are the KDs. I don't consider even hits, this 150 micromole. Agreed, this is a difficult problem, but when you use the smartest methods you can think, and the diversity of methods was very interesting. There were AI methods, there were also high throughput docking, like people using large servers to just dock. So some of these groups that were successful used a myriad of methods. So we don't have one method that now, no, we don't have AlphaFold. Nothing outperforms all the other methods, and it's amazing. No, this is the hit rate, different methods, and not that great. And then they asked them, the ones that succeeded, to expand the hits, yeah, into chemical series. Again, only 7 succeeded, and these are the affinities. That's the reality of hit finding for difficult proteins, yeah? So we have work to do, no worry, don't worry, guys. So how we think, and this is more forward-looking, that we can improve this, yeah? So we think we need more data, and you'll see some initiatives that are out there to generate tons of data for us to train. I think this is huge. We need to keep organizing these challenges because this is where we know how the algorithms really compare and where we learn how to improve. And we also think that we need to build an international community like with CASP. So in terms of data, this paper is coming up recently, but we've established a big consortium with pharma. And the idea is to screen with two technologies that enable us to screen large chemical libraries, more than 1,000 understudied proteins. So this is where, if you remember the human that we played, this is the top part, yeah? So we are gonna generate data in that part. I think this is really exciting. And what I find more exciting about this project is that these guys, they are experimentalists, but they are thinking in developing data to help us train better models from the start. I think this is really powerful. Well, again, this is a bit the roadmap, and there's opportunities as well if you have a protein of interest, but I don't think this is the audience. What else? Then I said challenges. So again, this is also a bit of advertisement, but if you are working in this kind of space, if you have docking methods of AI in virtual screening type of things, we are setting up a collaboration between CASP and DREAM. DREAM is also an organizer of computational challenges to launch over the next 5 years a lot of these types of challenges. There will be a main interest in hit finding, because what we want is to find hits for proteins for which we have no known ligand. But there will be other types of challenges as well, like post prediction, because these guys are crystallographers, so they will crystallize a lot of these hits to try to expand some of those series. No? So if you're doing more molecular dynamics to predict delta G, this type of thing, these sort of challenges could be interested. And again, we are very open to hearing about the community. So I was recently hearing that selectivity is really important. So we should do some challenges in selectivity. So we are thinking about that. And the first challenge, well, it's running, so you cannot join anymore. But I think it's interesting. We try to train models with DNA chemical library data. We use commercial DNA libraries. So it was a bit challenging because we didn't know the smiles of the hits, just the descriptors. So again, some of the classical deep learning methods, we couldn't use them, or we couldn't use them in the classical way. And we were asked to predict hits from another chemical library that was screened. I can tell you the first part of the challenge already finished. We were 50 teams. My lab also competed. And again, to be really honest, I don't think this is just about winning. So we didn't win. We were 10. I think it's quite decent. But again, like you saw in the challenge, the performance of all the methods was really low. Yeah. So we had decided to pick 20 teams, the top 20 teams for the prospective validation phase to buy new molecules, yeah? And in the end, only 8 teams had enough performance, and we had to lower the bar. So this tells you that we have a lot of room for improvement. We have a lot of things to learn. This is not solved, yeah? But I think we are going in the right direction if we understand what did the methods that perform the better do, and we can move forward as a community. And the last part is precisely the community. So I'm chairing this mainframe group. This is free to join. And basically you comply to open sourcing your methods and participating in some of these challenges. And then you receive some benefits. Like if you are selected, they will buy the compounds for free to test prospective your algorithms, for example. And yeah, this is a bit outdated. We are now at about 190 scientists from 43 countries, I think. So again, if this sounds of interest, join us. I think it's going to be a very interesting next 5 years or so. What more? Hit to lead. Okay. Normally, in drug discovery, when you have already identified the hit, then you start to optimize it. And this is, normally you use multi-objective optimization, because as you all know, to discover a drug or a clinical candidate, it's not just a potent compound, yeah? You need to find an equilibrium between opposing properties. You need compounds that are soluble but if they are too soluble, they will be excreted too rapidly. But if they are not soluble, they will not penetrate, yeah? You need your compound to be metabolized, because if compound is not metabolized, it's gonna accumulate. But if it's metabolized too fast, it's not gonna have an effect. So you need to balance a lot of these properties. So normally we have experimentally what we call the screening cascade, where you start to analyze these different properties, but the reality is that a lot of them you should optimize early on. And we're talking about potency, we are talking about selectivity. I'm a bit against this, but normally the selectivity here at the beginning is just checked against a very close analog of your protein of interest. Say, if we were developing a part 1 selective inhibitor, you would check part 2, yeah? Permeability and solubility, the MPK. And then as you progress, you go more into PK, safety, etc. So you also have at your disposal servers that predict many of those proteins. So here are 2 examples that I do recommend. And it's quite nice. Like, for example, here I put a small molecule, and you can see a lot of machine learning predictions of a lot of these end properties. But again, you need to understand that a lot of these were trained on preclinical data that might not readily translate into clinical safety, for example. But I think it's a good place to start. And let's talk, I promise I would talk to you more about HSP90. So again, thinking about if we should design selective or non-selective inhibitors, so something I wanted to study during my postdoc is how this polypharmacology or selectivity evolves across drug discovery. So this is luminespid. This is an HSP90 inhibitor that, as I explained you, was developed at the lab where I did my postdoc, and it failed in clinical trials. Actually, to be honest with you, they spin out this company. They created a company with this compound, and it was acquired by Novartis. They ran clinical trials, but the problem with this company is that it had a bit of ocular toxicity, which is not great. People lose sight. It was reversible, but that's not what you wanna, if you're taking a new pill and you stop to see, it's not very nice. And the efficacy was not better than other therapies that were developed at the time. So the compound did not progress anymore. But what I was interested in is that, so using computational methods that you'll see in a second, like we predicted that this compound, instead of only binding to HSP90, could also bind to some kinase of targets, yeah? It's very interesting because this was not optimized. I think the clinical program was perfect at the time. They did a selectivity profiling, as I told you, only with the clinical candidates, so they were not willing to optimize this a lot. But this program was relatively old, so they don't have the large kinase panels we have today. So I think they tested a dozen of kinases or so, and they found no off-target. So they said, our compound is clean. Is it clean? Well, with modern technology, we can screen a lot of more targets. So we did the computational prediction. And then we tested this experimentally, and we confirmed that the clinical candidate binds to several kinases. But the interesting thing is that our computational methods had predicted that the different, the heat and the leads would also bind to different kinases, and we were able to confirm this experimentally. So I like to plot this like this. These are kind of evolutionary trajectories similar of cancer evolution. And you see, for example, so the heat inhibits these 3 kinases. PIK1 might be familiar to you. Then the leads don't inhibit, for example, these kinases, and then they come back with the drug. You have NTRK, for example, here, that also it's available, it's inhibited by the first lead, but not by the second or the drug. So, when you don't optimize these things, polypharmacology can evolve in a bit of a, serendipitous way. So, remember when I said that people just check selectivity here? What we argue is that they should check selectivity earlier, because this way they would know if the hit or their lead is inhibiting other proteins, and they might to develop dual inhibitors for these proteins. For example, HSP90 inhibitors failed as the selective inhibitors, let's say. But I don't have it. But NTRK is here. NTRK is an approved drug. Right? You have chosen it before. NTRK. NTRK inhibitors become resistant as all selective inhibitors. But HSP90 is a protein that folds NTRK. So maybe a dual inhibitor of NTRK and HSP90 could slow down resistance and could be a better inhibitor for these types of cancer. But because they never checked, they never had the opportunity to develop this double inhibitor here. And then for those of you who selected PEAK1, that's why my selection was a dual inhibitor between PEAK1 and HSP90, because we have evidence that we can do it, and it might be beneficial to slow down resistance. Okay. Preclinical validation, and we are going towards less and less data, yeah? So more difficult to use computational methods to predict this. This is where projects start to fail. And this is a nice example. Again, this was a program that was run at the Institute of Cancer Research, and it's very nice because you see nice paper in Nature Chemical Biology, but this is a failed drug discovery project. And they did everything right. They developed a CDK8, CDK19 inhibitor. They couldn't find selectivity between the two, so it's a dual inhibitor. They did all the off-target validation. It's quite clean. They tested all the toxicity in mice and rats. Perfect. They went to dogs, and it's toxic in dogs. Who could have predicted that? Very difficult. But again, like showing you that a lot of things we don't know. Like, to be honest, I'm not sure what to make of this. They had two different chemical series, which is the recommendation, no? Because maybe one fails because of unexpected toxicity. They did all the effort to develop the other one, and it also had toxicity in higher animal models. So in principle, everybody accepts that it's toxicity target related to CDK8, CDK19 inhibition. So this target is dead. Nobody will put in the money to do this again. But there are some recent papers suggesting that this toxicity is off target. Again, who knows? Very difficult to prove. And this brings me to this last hands-on session of it. So there's this paper that I really like. But they basically did is that they chose a set of clinical candidates that they had failed in the clinic. And they tested them. Sorry. They CRISPR out, so they knocked down the protein that was supposed to be the target of those drugs, and they tested them in those cancer cell lines that they didn't have the target of the drug, and the majority of them still killed the cancer cell lines. So do we understand what the clinical candidates that we are putting in patients are doing? It begs the question, no? And it's this idea, no, that, so this is all their papers on drug target networks, but if you can see, most of the small molecule compounds that are the circles bind to more than one target. No? This is the polypharmacology I explained before, and it's very prevalent. But one big problem that we face to predict polypharmacology is that we don't have the data to predict it. No? So what we ideally would need to predict polypharmacology well would be having a lot of compounds tested against a lot of targets, and this type of complete matrices that would give us all the information, the true positive, the true negatives, so we would know. The reality is that this is what we have. If you go to Campbell, that's the data that you have. You have lots of compounds tested against very few targets. Yeah, most compounds in Campbell are tested against one target. And then you have very few compounds that are more comprehensively screened. But this is very difficult to build robust models with such imbalanced data. And this, if you flip this also makes you think, whenever you read a paper or you see a resource or whatever that says, this is a selective inhibitor of this protein, you always need to be very cautious because our knowledge is very limited. These two molecules could seem selective for this target if you just know this information. But if we had more information, maybe one would be cleaner than the other. And we never know, yeah, because nobody has screened a molecule against the 20,000 human targets. So we need to be aware of this reality. They are always properties that are related to this polypharmacology of target binding. We know, for example, that higher lipophilicity is more related to polypharmacology, to binding to multiple proteins. You don't want to develop very lipophilic molecules because they stick everywhere, basically. And there are a lot of methods that try to predict of targets of compounds. We recently reviewed this. So again, like the classical methods tended to use chemical information. This is what I used during my PhD, and target binding. So this would be information in ChemBomb. There are methods that also use 3D structures like docking, or Tarfius dock is a famous resource. And we see increasingly methods that use omics data for target prediction, network data. Clinical information is rarer, but there was a very famous paper using side effect similarity to predict of targets that I think it's very, very nice. And what we are seeing increasingly is methods that combine several types of information. I think this is the way to go. But again, like we need to be aware of the constraints, for example, of the chemical space. This is a big thing that we need to be aware of. We don't even know the size of the chemical space. Yeah. What does this mean? Like if you consider Campbell, for example, if you train a model with Campbell and then you predict another compound that it's not in Campbell, you don't know, it's out of your chemical space. Yes. So when you develop computational methods, you need to be very aware of how reliable are your methods to predict what you are trying to predict, because we can never build completely robust methods because we don't know how this chemical space is composed. And the biological space is a bit different, yeah, because we know how many proteins are there in the human proteome. But again, the data we have is also limited, particularly the chemical data. Again, to predict of targets, normally what classical methods use is describe the molecule with different descriptors and then compare it with machine learning to compounds in public databases, say Campbell, that are known to bind to a specific target. And this way, you can predict, for example, the affinity of this compound that's not in Campbell against all the targets that are in Campbell, for example. And this is a tool that you can use, although I think it was very slow. We tried it before, but we can try to see it. It's called the polypharmacology browser. It's quite nice. You have different methods here that you can use, trained with different descriptors, etc. And if you put a molecule of interest, you can predict different of targets for this protein. Yeah, you need to be aware as well. So my research is also trying to point out to people how reliable methods are, because I think this is very important when we use them prospectively. So this is the paper I told you before about off targets of HSP90 inhibitors. So in the bottom, you have the computational predictions for 2 HSP90 inhibitors. In green are the predictions that were correct. In black, the predictions that were not correct. So as you can see, much more black than green. Yeah? A lot of the things that we predict are not true. And on top, you see the real experimental results. Some of the more potent of targets we didn't predict. So you need to be aware of this when you use this method. So the last, a bit hands-on. Let's see if it works. We can try it together. But this is one of the inhibitors, if you remember, from this paper, that was found that when you remove the intended target, which is this kinase here, it still works. So we don't know how this compound works, and this compound failed in the clinic, so it's quite relevant. So if you can try to play with this a little bit and try to predict if this compound could be binding other targets, for example. Yeah, and I'll give you 5 minutes. I don't know how we are doing in time. We have half an hour, no?
We have 5 minutes.
5 minutes, okay.
Actually, have 2 hours for the.
Okay. Yeah. I'll be quick. Then, yeah, let's do this quickly because I think it was not working. Yeah, it's working. I think the quickest is to draw it, although it's a bit tedious, but. can also upload documents. Yeah, here in the bottom, you have the performance of different methods in terms of recall and precision. So you can choose. Again, you use one of these two that are involved. And you just click and it takes a few minutes to calculate. Yeah, let's just skip it. You'll have time to play with this later. So if we just continue, imagine you've done what I just did. So you probably found several other kinases, including, for example, EGFR. So this paper was published a couple of years after this initial paper, and they found that actually EGFR is involved in the mechanism of action of this inhibitor. So maybe the effects that they were seeing in the clinic were due to EGFR inhibition as well. No? And maybe if they had developed this inhibitor in a condition more related with EGFR, or what both targets were important, they would have had more success. No? So if we don't understand enough how drugs work, we can't develop them in the clinic in the ideal conditions, and therefore, if they fail, no, we don't learn. So I think this is really, really important. Okay, just the end, the clinical validation, the phases of clinical trials. But just to show you how, and this is relevant for the exercise that we'll do later. Sometimes, even today, we don't completely know what's best, yeah? And this is this story of CDK4/6 inhibitor. So it became clear that CDK4 and CDK6, that are very homologous, were important in cancer. So a lot of companies starting developing CDK4/6 inhibitor. Then palbociclib was approved as a breast cancer treatment in 2015, I think. And this was a revolution, right? Because breast cancer is really prevalent, so this had a big impact. And a lot of companies started to develop CDK4/6 inhibitors, and they are given, I think, in combination with hormonal therapy. But then now there is this trend of trying to give drugs earlier, yeah? So when they tried to give drugs earlier, palbociclib and other CDK4/6 inhibitor failed, but abemaciclib worked, yeah? So you have 3 approved CDK4/6 inhibitors, but in one disease context, only one of the 3 works, and nobody knew why. Basically, the developers, which was Eli Lilly, knew that abemaciclib was not as clean as the others, but they were hiding it, no? Because no, our compound is a selective CDK4/6. And then when it showed efficacy in this other context, they started to say, well, maybe it binds other CDKs as well, no? So it was not so clean. So again, this story. Well, I think I want to skip this story. This is kind of my research studying drug metabolites that I think is another area that we don't often consider, right? This talk is about having a systems perspective. We are always thinking about the drug, but all the drugs that you take are metabolized. And more often than you would think, there are metabolites quite concentrated in the plasma of patients that nobody pays any attention to, and that they might be doing something. And this is some research that we are doing at the moment. And again, this is some work trying to predict side effects. The amount of data that we have about side effects is limited because it's limited to the drugs or the clinical candidates that we have approved. It's about 4,000. So again, the models you can build are not as robust, but there's a lot of people working on this because it's very relevant if you could predict in advance the side effects. And that's it, guys. So I hope I have convinced you that this kind of how you should look to drug discovery, at least in oncology, is not a linear, simple process that it finishes when you approve the drug. It's the start when you approve a drug. And hopefully I have convinced you that things are much more complex. No, it's just not just a molecule binding to a specific target, but it's a molecule that it's metabolized, that binds to different targets and has different kind of impact depending on the people and the expression, etc. So I think reductionistic approaches are not enough to discover safe and efficacious drugs anymore. I think the easier diseases, the leukemia like imatinib diseases are gone. The diseases that had a single target that was the only responsible for that disease, we've solved. And now we are left with complex diseases where multiple targets are important. So we need this cycle of drug discovery. We need better data, particularly from the clinic, and use it more smarter with AI to try to discover drugs more quickly for patient benefit. And we need this systems perspective, I think, because being reductionistic is not going to get us very far anymore. And I find this optimistic. This is already a couple of years old, but this Nature editorial, I believe with it. I think that the AI revolution is not yet here because we don't have the data yet, but there are several initiatives, not just the one I showed you, but Google DeepMind announced recently that they also want to discover or put in the public domain a lot of crystallographic data of compounds and proteins to train algorithms. So I think the data is coming and probably the AI revolution in chemistry is coming as well. And that's it. Thank you very much. And I'll see you after the break.
Albert, if you want, maybe if you have questions, we can do the questions before the computer exercise because now we have the...
Yeah, or reach me in the coffee. And
let me say that you are not from Girona. I already said Girona because the next speakers in the afternoon, they are indeed from Girona. So Albert is from the Catalan Institute of Oncology and also Belvitge Biomedical Research Institute, right, in Barcelona. So sorry for that.
No worries. I was born in Girona, sorry.
Okay, so now we go for the coffee and then at 11:30 we are back for the computer exercise.
Pues pensaba que teníamos.
No, a 1:30 para llegar hasta la... Luego si tienes 2:30,
Bien. Creo que sí. A ver ahora la práctica. Claro, normalmente lo veo, pero después de ellos aquí no sé,